Hello, everyone, and welcome. My name is Rula Adib. I'm a senior principal at Geosyntac Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to advance the remediation of munitions constituents in contaminated surface and/or groundwater. First, Dr. Craig Tobias from the University of Connecticut will present his work to evaluate the natural attenuation of munitions constituents as well as a remedial option. Craig's presentation will be followed by a brief Q&A session. Then, Dr. Wenchang Zhu from Villanova University will discuss her efforts to optimize munitions constituents hydrolysis and adsorption by carbonaceous amendments. Wenchang's presentation will also be followed by a brief Q&A session, and we will conclude the webinar with a longer Q&A featuring both of our speakers. The next several slides provide instructions on optimizing your webinar experience. If you have not done so, please download Zoom at the link shown here and provide it to you in your registration confirmation email. If you cannot download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you have difficulties with your screen freezing, try keying in Control and F5 to perform a hard refresh. If you are accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to experience difficulties, please call into the conference line shown here. You can also submit a comment using the chat box, but please use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. And in case of continued technical difficulties, just download a PDF copy of the slides from the webinar webpage and call into the conference line provided to you in your registration confirmation email. Note that another option to view the slides or today's proceedings will be on the SERDP and ESTCP YouTube channel at the link shown here. Today's broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit them well in advance of the Q&A sessions. When you do submit your questions, please add your organization name at the end of your question so that we can identify it during the Q&A sessions. With that, I would like to introduce Cara Patton, who is an environmental project manager at Nobles and the technical assistant for SERDP and ESTCP's environmental restoration program area. Cara has worked with SERDP and ESTCP since 2008. And before that, she worked at the Drug Enforcement Administration, where she was a forensic chemist. Cara received her master's degree in chemistry from George Mason University. Cara, please proceed.
Thank you, Rula, and thank you everyone for joining the webinar today. First, I wanted to give you a brief overview of who we are, we being SERDP and ESTCP, before we jump into the presentations. We are two companion programs. The Strategic Environmental Research and Development Program is a partnership between the Department of Defense, EPA, and the Department of Energy, and is our traditional R&D program. We have been around since 1991, and our goal is to take our fundamental research use it to impact real-world environmental management, addressing real-world issues and managing real-world issues at our installations. SERDP operates in companionship with ESTCP, or the Environmental Security Technology Certification Program, which is a demonstration and validation program operated out of the Department of Defense, which is designed to take research, knowledge, and technologies developed under SERDP and other research programs and demonstrate them in the field to get a good cost and performance data at a pilot scale level to help when we want to get to full scale, with the ultimate goal of being technology transfer of these technologies to accelerate their broader application and commercialization. There are a few environmental drivers that guide our research and demonstrations that we fund. These very broad issues are applicable to many DoD installations, anything from maritime sustainability to climate issues, noise, UXO, and munitions constituents. We are also looking to reduce current and future liability, which is also quite broad and can take the form of pollution prevention to eliminate hazardous materials or processes at our military installations, and issues from past practices where we have had impacted groundwater, soils, and sediments, and some emerging chemicals of concern issues. We have a number of focus areas. You can see from this list that they are very broad. From the top of the list with PFAS down to electrical infrastructure and planning. The presentations today will be discussing munitions remediation. It is very important for us to transfer the information being developed under SERDP and ESTCP, and we have a number of ways that we do this. Through in-person trainings, videos, guidance, and ultimately our webinars. We're always trying to get this information out to people that are interested in and will use the information. Shown here is a list of some future webinars. Our webinar series highlights research and demonstration efforts from all of our program areas. Upcoming topics include forecasting dryland ecosystem vulnerability to change, modeling and monitoring tools to support passive and active NAPL remediation approaches, as well as some others. Registration is open for webinars through early next year. You can register for these upcoming webinars at this live link shown here, and you will also be able to access archived webinars at this link as well. Registration is now open for the 2022 SERDP and ESTCP Symposium, which will be held November 29th to December 2nd in Arlington, Virginia. This event will showcase the latest technologies that enhance DoD's mission through improved environmental and energy performance. Finally, I would like to remind you that a copy of the presentation of today's session can be downloaded from our webinar webpage. We would appreciate it if you can please take a moment to complete the survey that will pop up on your screen at the end of the webcast. I hope you enjoyed today's webinar, and I will turn things back over to Rula to introduce our next speaker.
Thank you so much, Cara. It is now my pleasure to introduce our first speaker, Dr. Craig Tobias, who is a professor in the Department of Marine Sciences at the University of Connecticut. Craig has served as the principal investigator and co-PI of projects funded by SERDP, the National Science Foundation, the Environmental Protection Agency, and others, and has been widely recognized for his excellence in research and teaching. Craig's research portfolio spans the broad topic of pollutant fate and transport along the aquatic continuum from watersheds to the continental shelf. His work specifically targets agricultural and urban-derived contaminants, as well as legacy and next-generation munitions constituents in both surface water and groundwater. Finally, Craig received a bachelor's degree in biological sciences from the University of Delaware and a doctoral degree from the College of William and Mary from the School of Marine Science. Craig, welcome. Please go ahead and get us started.
Thanks for the introduction, Rula. Good afternoon or morning to everyone, and thanks in advance for your time. I'm going to spend the next 20 minutes or so talking about some enriched stable isotope approaches that we've used to get a better handle on constraining the amount of munitions constituent transformations, principally in surface waters, but also in groundwater environments. So this is the roadmap for my talk here. And as I go through it, what I'd like you to do is just keep in mind this underlying theme that all of the different aspects of this work that I talk about are really designed to help quantify or get better estimates of what munitions constituent transport and specifically losses are under pretty realistic conditions across a range of different kinds of environments that you're likely to encounter. So I think that if you're sitting in on this seminar, you are acutely aware of the fact that DoD has several installations where there are regulatory decisions that are being executed based upon munitions constituent contamination, either in surface waters or in groundwaters, or in some cases both, because of the connectivity between the two. So these munitions constituents presently largely consist of legacy compounds that have been used for, well, close to 100 years now, that have resulted from principally manufacturing, storage, and handling, and perhaps to a lesser extent, their use on test ranges. But what we're also seeing now is we're seeing an increased use of insensitive high explosives, often referred to as IHEs, that are becoming more increasingly used in munitions constituent formulations. So we're likely to start seeing some of these contaminants emerging, not so much probably from manufacturing, storage, and handling, but more likely from migration offsite of test ranges. So here are the cast of characters that I'm going to touch on at various points during this talk. So the two legacy compounds that I'm going to focus on here are RDX and TNT, the primary constituents found in composition B, which is widely used in a variety of applications. And then the two IHEs that I'm going to talk about here are NTO and DNAN. And what's on this slide is a list of just very general sort of compound characteristics. And I think that there are some generalities that you can make about both IHEs and legacy compounds, in that relative to a lot of other organic contaminants, these compounds are relatively polar, maybe a little bit less so for the two nitroaromatics, DNAN and TNT. But in general, this relatively higher polarity means that they are more likely to migrate in surface water and groundwater environments. Now, in doing so, all of these compounds will undergo certain amounts of degradation and transformation. And many of these, or all of these compounds will yield multiple organic derivatives that come in a lot of different flavors. And so some of these flavors of organic derivatives here are listed in the central column of this table. And I don't want to get into the detail of these too much, but I do want to say that traditionally, one of the primary routes of evidence for compound degradation in the environment has been the presence of these organic derivatives. And what we've done now in really two SERDP projects and a NESD project was steer away from this more derivative approach and instead focus on complete mineralization of these compounds. So as these compounds break down, often these derivatives are stepping stones to complete mineralization, wherein that the carbon atoms that are contained in these compounds end up as CO2, or in some anaerobic environments as methane, but generally considered part of the DIC, or dissolved inorganic carbon pool. And the nitrogen atoms that are contained within these compounds, because these are nitrogen-rich compounds, will end up as dissolved inorganic nitrogen. And that can take the form of several different species. So either nitrate or nitrite, ammonia, dissolved nitrogen gas, nitrous oxide, really depending on the redox environment in which these compounds were completely mineralized. And so this is where our work focuses really, is it focuses on these mineralization products. And the reason for this is that even though the derivatives do provide some indication of munitions constituent breakdown, they, one, don't indicate on their own complete mineralization. And two, many of these derivatives retain some additional toxicity and have their own regulatory thresholds. So for instance, one of the breakdown products for DNAN under certain conditions is dinitrophenol, which actually has a higher toxicity than the parent compound itself. So by going after the mineralization end products, what we're after is being able to demonstrate how much of these munitions constituents were fully decomposed into inert products, those DIC and DIN species, because those have absolutely no toxicity associated with them, and thus no additional human health or ecosystem risk. Okay, so the rub here is that these DIN and DIC products are ubiquitous in the environment. So being able to specifically quantify the amount of DIC or DOC that is uniquely derived from the mineralization of munitions constituents is a very small signal against a very large background of noise. And this is where the stable isotope tracer techniques come in, because it allows us to uniquely differentiate the mineralization products that were derived from the decomposition of munitions constituents from all of the countless numbers of natural sources that are producing the same thing. And so the way this works is that we first have these parent compounds synthesized where we substitute what I'm going to call normal carbon or nitrogen atoms, so C12 or N14 atoms. We substitute those with their enriched isotopic counterparts. So DNAN, for instance, could be synthesized with C13 being used to form the nitro, or to form the aromatic ring. RDX, for instance, can be synthesized where you have use N15 atoms for the nitro groups or the ring. These compounds can be synthesized with either C13 labels in different positions on the molecule, or N15 in different positions on the molecule, or dual label when we've used both dual label sources of MC compounds, and we've also used single label. Okay, so once we have these isotopically labeled compounds, we now have the ability to be able to trace these specific carbon or nitrogen atoms as they're mineralized into the DIC or DIN pool with a sensitivity of now that's about 1 in 100,000. So that signal to noise problem that I mentioned in the previous slide largely goes away. We can now uniquely identify the amount of mineralization products that come uniquely from the mineralization of MC. So we have these labeled compounds, and what we do is we use them in a series of experiments where these experiments are designed to simulate natural habitats. And we do this across a range of habitats. So we might have experiments that are running just sand, sediment, or silt, or one aquaria with aquatic plants in it, like we have here. Or sometimes what we do is we'll do very large mesocosms or ecocosms, like we have here, where we constructed an intertidal wetland that we can manipulate the ecohydrology in. and get it to flood and drain and flood and drain like you would see in a natural environment. So these experiments have, sort of quasi-natural habitats that now have these labeled compounds in them. And then over time, what we can do is we can look at the rate of incorporation or the production of the isotopic label, either C13 and DIC or N15 and DIN. Over time, that allows us to calculate that unique, that MC unique mineralization rate, and we can derive rate constants. Okay, so we do this across a range of aquatic habitat types, and we basically get 4 pieces of information out of this, and that's what these bullet points are. So we get rates of compound loss from the aqueous phase, how much of that parent compound actually goes away. Then we can attribute how much of that loss was specifically to absorption onto sediments. So just the physical partitioning, no biology, no degradation. And then uniquely using the isotopic label, we can quantify how much of that loss is attributable specific to the mineralization of that compound. Right, mineralization is what we want. That's the brass ring for degradation. And then lastly, because we do this across a range of environments, we can start looking at what environmental characteristics might actually be important for controlling the rate of or the amount of absorption, or more specifically, the amount of mineralization. Okay, so what I'm going to do is for the next 4 or 5 slides, I'm just going to give you a data example from some of these experiments that correspond to each one of these bullet points. Okay, so the first one is just some experiments we did with NTO. Each one of these series represent a different quote unquote habitat type. So we have sand, silt, we have wetland, we have one where we added plants. That was that picture on the last slide. And then we have a couple where we've amended these with natural sources of organic matter, one with leaf litter and one with phytoplankton. And so this plot is just looking at the disappearance of the parent compound over time. And you can see that there's quite a range of that loss depending upon, what the characteristics of the habitat type are, with some of our higher rates happening in the organic amendments or where we have a lot of aquatic plants. Okay, so we know how much of that compound is going away from the overlying water column. To determine how much of that is due to just physicochemical absorption, what we can do is we run sort of parallel experiments, which are very similar to classic absorption isotherm experiments, where we run them under sterile conditions with gamma irradiated sediments and filter sterilized overlying water. And we also run a parallel with sort of unsterile conditions too. And here's, some absorption curves for an experiment that we ran using DNAM. And ultimately what we get out of this is we get sort of very classical parameters in terms of absorption coefficients and partition coefficients. And these are very critical parameters that go into building fate and transport models. So essentially what we're doing is we're starting to compile like a menu of rate constants across different kinds of habitat types to constrain these models. So here's a data example where we're using the isotopes to derive how much of that fractional NTO loss that we saw 2 slides ago is actually due to NTO mineralization. And that's derived from the amount of isotopic label, in this case 15N, that we were able to measure being accumulated in the DIN pool. And so again, different habitat types, sand, silt, plants, wetland environment. And what you can see is that there's a very, very large range in the amount of mineralization that's occurring as you move through a relatively organic matter poor habitat to a relatively organic matter rich habitat. Okay, and so here we're deriving some rate constants, but these are now unique to mineralization, as is the half-life that we've derived from those constants. Because we do these across a range of habitats, we can start exploring what factors go into making that very large range of loss or absorption or mineralization. And we do this in a couple of different ways. Some statistical approaches that are relatively straightforward and some statistical approaches that are maybe not so straightforward. And the way we start is we start with a multivariate analysis done in the lower right-hand corner as a principal components analysis. And effectively what this analysis does is it lets us look at all of the environmental parameters and figure out which ones are potentially important for explaining the variance that we see in some of these rate constants. And then once we've identified some of those specific environmental factors, what we can do is then, so this top plot actually shows some rate constants for DNAN across a range of increasing organic carbon. We can then take and we can derive mathematical functions based upon what those environmental parameters are. So essentially we're looking for environmental proxies that correspond to whether or not you're going to have a high mineralization rate or low mineralization rate. And these kind of proxies, again, are very useful for constructing reaction and transport models and parameterizing them. And then so lastly, because we look at these different factors, we're starting to run experiments now where we're trying to nudge specific habitats or ecosystems in a direction that might enhance mineralization or MC degradation. And so in the bottom, these pie charts represent some work that we did with RDX. And the gray area of these pie charts corresponds to how much RDX was truly mineralized. And what we saw was that by manipulating mesocosms through a period of flooding and draining and flooding, what we were able to do is cycle through high redox, low redox, high redox, low redox, and that translated into an increased rate of RDX mineralization. The other kind of manipulation that we're doing, and we started this with the IHE work, is trying to use natural sources of organic matter to enhance mineralization. And so here's representation of the NTO mineralization relative to our lowest rate, which was in sand, right? And you see under natural conditions, the higher organic matter environments have higher mineralization. But it turns out that if we go back to, plain old, relatively unreactive sand, and we add leaf litter or we add phytoplankton, microalgal derived organic matter, we can get almost a 10-fold bump in the mineralization rate. Okay, so we can start thinking about how we can apply this to different sites, right? So here are some of the benefits that you get from the work that we've done, constraining these reaction parameters, providing some additional evidence for natural attenuation. But let's just think about a couple of hypothetical or not so hypothetical examples. So I'm thinking of a non-hypothetical example of an RDX contamination in a stream that flows for kilometers. And it goes through different parts of the stream, pools and riffles, spends some time in some ponds where you're likely to have higher organic matter and a higher resonance time. Each one of those sections of that travel path has different mineralization or loss rates. And the kind of work that we're doing can assign or ascribe specific kinetic functionality to each of those reaches. So that helps you understand better how that compound is behaving during transport. This figure right here is actually a map of an RDX contaminated groundwater plume at a site where we use this approach where we amended this plume with N15 labeled RDX and use that to calculate the mineralization of RDX actually in the aquifer and relate that to what the transport time was to determine whether or not natural attenuation was a reasonable alternative for this site. So if you are an RPM and you're working at an MC contaminated site, you or your contractor is undoubtedly familiar with this reaction and transport equation, right? You have some transport terms in here. And two of these terms that are particularly difficult to parameterize in models are the adsorption term and definitely this reactivity term. And the kind of work that we're doing is providing you, again, sort of a menu of parameterizations that you might use depending on what your habitat type is. So this slide here is actually part of that groundwater study that we did in the RDX contaminated aquifer. This cattle tank has groundwater that has been amended with N15 labeled RDX. And what we did was we used this to provide some additional evidence of complete mineralization of RDX half-lives under both anaerobic and aerobic conditions. And so this is important because, you know, regulatory agencies, in my experience, for natural attenuation to be considered, you're looking for multiple lines of evidence. They're looking for loss of parent compound, they're looking for appearance of derivatives. But to be able to go in and say specifically, quantitatively, this is how much is getting mineralized under natural conditions is, you know, potentially a powerful third leg for that stool. One other possible application of this, which I'm particularly excited about, is trying to put nature to work. And it's basically, can we manipulate some sites or create some eco-type treatment to help remove MC compounds? You know, whereas, you know, can we manipulate the eco-hydrology, like in this pie chart example that I referred to earlier? Or, you know, can we use wetlands and perhaps make some modifications to wetlands to really enhance the amount of natural organic matter that's there to increase the removal rate? Orange County, California uses treatment of wetlands to remove pesticides from water that's destined for aquifer recharge. South Florida Water Management District does it for nutrient removal. Savannah River site does it to remove tritium, of all things. So I think that this potential application is really underutilized, I think, for MC. And our work helps to constrain the efficacy of that approach. All right, so here are the conclusions. I've kind of hit on these throughout the talk. You can know what the mineralization rate of these compounds is using this approach across a wide range of habitats, and it works in situ in aquifers. You can take the information, you can plug it in to help parameterize fate and transport models. There is the strong potential for putting nature to work and manipulating ecosystems to help you amplify natural attenuation. And the one thing that I'm interested in exploring in the future is that we've only done this approach with MC compounds, but this approach should work with other contaminants, as long as those contaminants can be synthesized with a stable isotopic label. And I think all of this can be distilled down to some benefits to DoD, which is, you know, additional evidence to support whether or not natural attenuation is a viable option for your particular scenario. Because certainly if it is, the implications are, you know, lower cost, lower effort, with the same or potentially enhanced efficacy for removing and degrading MC constituents. So with that, I will end. Thank you for your time.
Great, thank you so much, Craig. As a reminder to our audience members, please submit your questions using the Q&A box on your screen. We have received several questions that Craig will answer. The first one is from the North Carolina Department of Environmental Quality. Did you conduct any desorption experiments? And if so, did you observe any hysteresis?
So desorption experiments. Well, we've done a lot of sorption experiments. I think maybe what the question is asking is, after we introduce the labeled compounds, do we see them linger? And then consequently, are they exchanged and desorbed over time? That is all part of sort of the time series data that we collect during these experiments. So these experiments, we've conducted them as both pulse experiments, and we've also conducted them as steady state type experiments. And we typically run them for 3 weeks to a month. So really, over that time series, you have a lot of things that are changing. And then those are all accounted for when we end up modeling the rate constants. For the in situ work, when we added the labeled RDX into the aquifer, what we did was we followed that work with a very, very aggressive pump back period, so that any of the unreacted labeled RDX was sort of harvested back from the aquifer. So I hope I understood your question correctly. If not, I'm happy to address it, offline. You feel free to email me.
Great, thank you. A follow-on question, also from the North Carolina Department of Environmental Quality. Do you have insight if the data you showed or the experiments you conducted, if the sorption mechanisms are reversible or irreversible?
Yeah, that's the answer is yes. So let me clarify. It depends on the compound. So certainly one thing that we do see for TNT, and a lot of other people have noticed this, is that if you get some reduction of TNT to TAT, which is triamino-toluene, that tends to bind irreversibly to the sediments. And that is a big contributor for why TNT can be, and its derivatives can be so persistent in the environment. For the non-nitro aromatics, and we're just looking into the data for that for DNAN as well. So DNAN, the nitro aromatics tend to be much more sticky as a function of both organic matter and clay content. For RDX and certainly for NTO, sorption is much less of an issue. So I think it's coming and going. We do not specifically measure how the C13 or N15 enrichment is evolving over time, because the way we do these experiments is we do them in non-contaminated settings, environments, with the exception of the groundwater environment. So certainly the irreversible absorption is more of an issue for the nitrate aromatics. For NTO, it's so polar, we really can't even get it to stick unless we have really, really high organic matter content. And even then, I think part of that is degradation.
Thank you, Craig. This next question is from the U.S. Army. Is there a vapor intrusion issue associated with any of the munitions constituents that you studied?
That is not my area of expertise, but I would venture a guess to say no. Right? These the vapor pressures for these compounds are very low. Like when we started our initial work with the legacy munitions, we explored whether or not we needed to account for gas exchange of the parent compounds or its derivatives when we were doing our isotope mass balances at the end of the studies. And our conclusion was that it was trivial. It was so small we didn't account for it.
Thank you so much. Another question from the U.S. Army. For the site that you described, where can the details for the approach and the results be found?
Oh,
Have you
yeah,
published
great.
any paper, or are they available in your final report, and how can we find it?
Yeah, that's a great question, and I meant to give a plug for that. That was for the, and I assume the question is about the in situ groundwater experiment. That was part of NESD project 544, and there is a final report for that. For all of the other mesocosm and aquarium scale work that I presented, for the legacy compounds for RDX and TNT, that is in a prior SERDP final report, which is, I can get you the actual project number for that. I forget it right now. And then for the legacy, or for the IHE work that we're doing presently, that project is still ongoing. And so that final report will be submitted by July of 2023. And that project number is ER 19-1054.
We'll put the links for these in the chat, or
Yeah.
if you can do it, Craig, that would be great.
Yeah, perfect. And if you need that,
After the
that
Q&A
other,
session.
yeah, if you need that other project
Yeah.
number, just let me know.
Okay, great. Yeah, if you could just put them in, and we'll look up the links and add them, if you can put in the project numbers. But
Sure, yeah.
let's go through a few more questions before we move on to our next speaker. This next question is from the University of Tübingen in Germany. Did you measure incorporation of these compounds into microbial biomass, if any, over time?
Oh, yeah, that's a great question. Yeah, so the work that I did on that NESD project, yeah, Tom Boyd was actually doing some parallel work. I worked with Tom on that, and we did a couple of injections of data that I did not show, where it was dual labeled C13 and N15 RDX under biostimulated and bio-augmented in native conditions. And in the bio-augmented ones, so those actually, that was, the injection had cells of cultured known RDX degraders. What we did find was that some of that label for both C13 and N15 was incorporated into biomass. And what happened was, over the next year, both labeled DIC and labeled ammonia sort of slowly bled out of that biomass. So our conclusion, and that is in a ES&T paper that's almost ready to submit. And the conclusion from that was that the amount of the tracer that was incorporated into biomass actually equates with mineralization, right? Because it is slowly being mineralized and leaked out over time. So if you calculate the mineralization rate from just the amount that you see in DIN and DIC while you're doing the experiment, it's actually a conservative estimate of mineralization. But we only saw that in the bio-augmented treatments. So I think there is some incorporation, but it only becomes very important to account for if you are adding cells to your experiment or to your groundwater. Yeah, good question.
Great, thank you. We're gonna try and get to the remaining questions for you towards the end, but at this point, we'd like to thank you for a great presentation and introduce our next speaker, Dr. Wencheng Zhu, who is an associate professor in the Department of Civil and Environmental Engineering at Villanova University in Pennsylvania. Wencheng's research aims to better understand the various ways that nature detoxifies contaminants by the integration of chemical, biological, and material science approaches to attenuate pollutant transport, thereby protecting source water, and to degrade contaminants with novel materials to provide safe drinking water. Wen Cheng has served as a principal and co-principal investigator of projects funded by SERDP, NSF, NIEHS, and EPA on a number of pollutants, including chlorinated solvents, pesticides, munitions constituents, PFAS, and disinfection byproducts. She is the recipient of various awards, and she received a bachelor's degree in environmental engineering from Nankai University in China, a master's degree in geography and environmental engineering from Johns Hopkins, and a doctoral degree in chemical and environmental engineering from Yale University. Wen Cheng, we're happy to have you. Please proceed.
Thank you so much for the introduction, Rula. Hello, everyone. It's a great honor to be here. Today, I'm going to talk about our SERDP project, optimizing carbon emittents that simultaneously adsorb and transform legacy and insensitive high explosives. This is an agenda for today's talk. In this talk, I'll focus on two chemicals in munition constituents, namely DNAN and NTO, because they're widely used in insensitive high explosives, IHEs. Typically, they could accumulate on DoD ranges as post-detonation residuals when low-order detonation occurs. And these areas can subsequently serve as source zone for groundwater contamination, because many of these compounds are highly water-soluble, and some of them are charged. For instance, the compound I showed on the slide, that is NTO, which carries a negative charge under environmental relevant conditions. This makes it very easy to penetrate through soil column and into groundwater. As a result, there's an urgent need to develop a technology that can simultaneously retain and also transform IHEs. Among all the technologies to retain IHEs, pyrogenic carbonaceous matter, which I'll refer to as PCM, it includes activated carbon and biochar, is a promising material because it has the demonstrated effectiveness as a passive absorbent. For instance, several SERDP projects have shown that activated carbon, AC, can be used as emittents to strip PCBs from pore water and ultimately reduce their toxicity. AC has also been used to scrub wastewater IHEs. However, the main challenge of PCM is that it is ineffective at retaining highly water-soluble IHEs, such as NTO. It also requires AC disposal or regeneration once the absorption sites are saturated, which can be costly. When it comes down to IHE destruction, the most commonly used method is alkaline hydrolysis. This method is easy to implement, and the material, lime, is very cost-effective. In addition, alkaline hydrolysis has the demonstrated effectiveness towards RDX, TNT, and DNAN at both lab and field scales through the support of SERDP ESTCP projects. However, the challenges remain in that alkaline hydrolysis requires extreme pH values. It often has to go above pH 11 to work. Under such extreme pH conditions, it also often mobilizes metal co-contaminants. Moreover, it is ineffective for some of the IHEs, such as NTO. Another method that could potentially be used for IHE destruction is in situ chemical oxidation with peroxydisulfate, which I'll refer to as PDS. PDS is the most powerful oxidant in the peroxygen family. It also has been applied as an in situ chemical oxidation technology for groundwater remediation, although its effectiveness towards IHEs are yet to be demonstrated. So in this project, our goal is to develop a technology using optimized PCM to enhance IHE removal. Specifically, we will identify critical properties of PCM and then populate those properties to promote simultaneous IHE adsorption and destruction. Today, I'll focus on two aspects of our ongoing efforts. One is the using PCM to simultaneously adsorb and promote hydrolysis of DNAN. The other aspect is using PCM to promote the simultaneous adsorption and oxidation of NTO. So let's take a look at DNAN first. We have so far demonstrated that PCM can accelerate DNAN hydrolysis. As shown in panel A here, DNAN decay demonstrated first-order kinetics, where the slope represents the rate constants. The empty circles are DNAN hydrolysis without PCM, whereas the solid circles are DNAN hydrolysis with PCM. As you can see, the addition of PCM was able to shorten the half-life of DNAN from 17 to 9.5 days. As shown in panel B, as DNAN disappeared, we observed the formation of nitrites in green and 2,4-dinitrophenol in purple. So how did PCM promote DNAN hydrolysis? To answer this question, we performed kinetic experiments at 3 different temperatures to obtain the activation energy for DNAN hydrolysis with or without PCM. The solid circles here are DNAN decay with PCM, whereas the empty circles are DNAN decay without PCM. The slope, according to the Arrhenius equation on the right, indicates the activation energy for the reaction. As you can see, our results suggest the presence of PCM was able to decrease the activation energy significantly by about 54%. So the next question is, what PCM properties are important? As shown in this little cartoon here, PCM is rich in chemistry. First of all, it has various functional groups on its surface. The phenolic group, in particular, has been proposed to act as a weak base and therefore promote alkaline hydrolysis for certain compounds. PCM also has different pore characteristics, ranging from micropores, those are below 2 nanometer, all the way to macropores, those are above 50 nanometers. Recently, there has been a lot of attention on the micropores due to their ability in accelerating reactions by nanoconfinement. However, the challenge is that these properties in PCM are intrinsically linked, so it is very difficult to study them individually. To address this challenge, we developed a tunable PCM-like polymer networks, which I'll refer to as PLP. This allows us to investigate functional group identity and pore characteristics individually. Like PCM, PLPs have large surface area, high microporosity. They're also highly conjugated and amorphous. Moreover, they have superior affinity towards apolar contaminants. But unlike PCM, PLPs are highly tunable. For instance, we can incorporate specific functional group through the selection of R group in the linker through the polymer synthesis. We can also control the pore characteristics through the selection of monomer struts. So in this work, we synthesized 4 PLPs on the left with different functional group identity. OH group with quaternary ammonium group, which I'll refer to as QA. NH2 group, and tertiary amine group. We also synthesized the 2 PLPs with different pore characteristics on the right. Specifically, the PLP micro has a mixture of micropores and metapores, versus the PLP metal has no micropores. The reactivity of all 6 PLPs are then monitored by the decay rates of TNT, which is a legacy explosive, and it is also a structure analog to DNAN. Both the functional group and pore distribution are confirmed, and the results are in my backup slides. So we first monitored TNT decay rates in the presence of the 4 synthesis of PLPs with different functional groups at pH 7. No TNT decay was observed at pH 7 without PLPs. By contrast, the addition of 4 PLPs here accelerated TNT decay to different extent. The slope here represents the rate constants. As you can see, the fastest is the PLP with OH and QA group, followed by NH2 group, followed by OH group, and the tertiary amine group showed very little effects. This can be explained by the basicity of the functional groups generally. Another interesting observation, as shown in panel B, is that by simply crafting QA group onto PLP-OH, as shown in the red versus the black, you can see the reactivity of PLP increased by 4 times at pH 7. The enhancement became more evident at higher pH. For instance, we observe an enhancement factor of 10 at pH 9, which yields a half-life of TNT within 1 day. So we hypothesized that the observed enhancement was due to the ability of QA group on PCM to attract and accumulate OH-, which resulted a high local pH for alkaline hydrolysis. We will further verify this hypothesis in the modeling parts. We also compared the reactivity of 2 PLPs with different pore characteristics. The brown circle represents PLP with micropores versus the green triangle represents PLP with no micropores. Our results clearly indicate that the presence of micropores accelerated TNT decay, and the formation of nitrite, which is shown on the y-axis, on the right y-axis, mirrored the same trend. We then performed non-reactive molecular dynamic simulation to understand the mechanism. So here, we used 4 layers of graphene, which are shown in the black dots here, to represent a simple pore geometry of PCM. The distance between graphene layers were kept at 4 nanometers. We then filled the space with electrolytes, where red represents the water oxygen, blue represents the hydroxide oxygen, and green represents TNT. In panel B, we decorated the graphene sheet with QA groups, which are shown in purple. Chloride was then added into the system to keep the system charge neutral, which is shown in orange. There are several interesting observations here. First, TNT initially lied flat on the plain graphene sheets. However, as the QA group increased, TNT started to get pushed off the wall. At the same time, we observed that the OH-, the green dots, started to migrate near the wall. We then plotted the density profile in panel C and D, where OH- was shown in blue and TNT was shown in green. Our results suggest that at intermediate QA coverage, OH- and TNT were accumulated in the same spatial region, which could explain the enhanced hydrolysis we observed in our experiments. So, taking advantage of what we have learned from the polymer network, we then used a QA polymer, polydetmec, to modify activated carbon. Polydetmec is widely used for water treatment as a coagulant and is considered non-toxic. Here, we adsorbed it to pristine AC to place the QA group on PCM surface. We then monitored their performance in promoting hydrolysis. First of all, we looked at TNT here. The left figure is modified AC, and the right figure is the pristine AC. Our results suggest that the modified AC was able to speed up the reaction, and the half-life of TNT was shortened from 1 to 0.2 days. At the same time, we saw an enhancement in the nitrite formation. We then looked into their performance in promoting DNAN hydrolysis. Similar results were observed, although DNAN degradation proceeded a lot slower compared to TNT. But still, we were able to see that modified AC was able to shorten the half-life of DNAN from 19 to 13 days. And at the same time, it produced the nitrites, which indicate a near complete denitration. So that's our story on DNAN. Now I'm going to switch gears and talk a little bit about NTO. First of all, it is very important to point out that despite the fact that NTO is negatively charged and is very polar, and AC surface is negatively charged too, but under environmental condition, NTO has exceptional affinity towards activated carbon, as evidenced by its high KD value listed here. So why is that? We hypothesized that NTO may form a charge-assistant hydrogen bond with PCM. Specifically, if you're looking at NTO here, it has a pKa value of 3.76, which is close to the pKa value of carboxylic acid group on PCM. That means NTO and carboxylic acid group can share a proton equally, forming a very short but very strong charge-assistant hydrogen bond. To test our hypothesis, we conducted competitive absorption experiments using 3 different adsorbates. And NTO in this case was preloaded onto activated carbon surface. So the first adsorbate was p-toluene sulfonic acid. The chemical structure is shown here. It is incapable of forming charge-assistant hydrogen bonds because its pKa value is too far from the pKa of NTO. So as expected, as we add more p-toluene sulfonic acid into the system, it did not impact the adsorbed NTO. The second adsorbate was methyl benzoate, which is also incapable of forming a charge-assistant hydrogen bond because it has no acidic proton. So as shown in the red circle, methyl benzoate was not able to compete for absorption sites with NTO. By contrast, the addition of methyl benzoic acid was able to displace NTO from the absorption site, which means that this methyl benzoic acid can form a charge-assistant hydrogen bond. So another piece of evidence we have gathered is the distribution coefficient of NTO over pH range. As you can see in this figure, the black dots represent the experimentally measured KD values of NTO. We then used the 2 different models to predict the KD values. One is linear speciation model, one is non-linear speciation model. I'm not going to go into all the details here, but as you can see that our measured KD values clearly deviates from both models. And the deviation is represented by this big hump in the data and the positive shifts in pKa values, which confirms the formation of a strong charge-assistant hydrogen bond type of bonding between NTO and the surface acidic groups of activated carbon. And this is very important because we can then potentially populate the surface with more carboxylic acid to better retain NTO. Now that we have demonstrated that PCM can adsorb NTO favorably, we then added a small amount of peroxydisulfate, PDS. And what we found is all the NTO in the system were completely destroyed within 1 hour at pH 7. This is very important because it suggests a potential route for material regeneration, and then we can recycle the GAC. We then carried out some preliminary work to evaluate the stability of activated carbon in NTL degradation. Specifically, we added 10 cycles of sequential NTL to GAC, followed by PDS addition. The bar in the shaded area is the control in the absence of GAC, where we see no NTL degradation by PDS. On the right, it is the 10 cycles of NTL decay in the presence of PDS and GAC. We can see that GAC generally maintained its performance with a slight decrease in reactivity towards the very end. And overall, GAC was able to degrade 28% of its own weight after 10 cycles at neutral pH. So to conclude, in this project, different from the conventional wisdom where PCM is inert, passive absorbent, we have demonstrated the dual role of PCM. Specifically, PCM was able to simultaneously concentrate and promote the decay of both DNAN and NTL. We also use the fact that the absorbed NTL and DNAN can be destroyed on the surface suggests a very important route for activated carbon regeneration, which is attractive to engineering application down the line. We also use a polymer approach to identify the critical properties for PCM in this process. And these information will be fed to our future work, where PCM will be optimized to populate those properties and enhance its performance for IHE removal. Once we obtain and optimize the PCM, we will then evaluate its effectiveness towards post-detonation residuals. In terms of the benefit to DoD, this project will provide critical knowledge on tailoring PCM that can be used as range amendment to simultaneously retain and destroy legacy and IHEs. Depends on the form of the amendments. It could also be tuned into a more injectable form for possible groundwater remediation. There's one aspect of this project I didn't get the chance to talk about is the computational side. We're currently in the process of developing new computational models, including one quantum chemistry model and a machine learning model, to help us predict products and reaction pathways for hydrolysis in heterogeneous systems. Previous work has been exclusively focused on hydrolysis in homogeneous system. So with that, I want to thank my team. The results I presented today are mostly from my team and Dr. Joe Pignatello's team from Connecticut Agriculture Experimental Station. I also listed several publications from this project as additional sources in case anyone wants to take a look further. Last but not least, this is the project information and this is my contact information. So I guess I'll end here and I'm happy to take any questions from the audience.
Thank you so much, Wencheng. We did receive several questions. This first one is from the US Army.
Mm-hmm.
Can you elaborate on the fate of the 2,4-dinitrophenol formed after alkaline hydrolysis of DNAN?
So that's a really good question. So the focus of this project is not necessarily on the environmental fates, but I would say that I'm aware that 2,4-dinitrophenol is more toxic than its parent compound, DNAN, in this case. So once it enters the environment, there could be other process that could facilitate its potential decay. For instance, the micro organisms and potentially abiotic reduction reaction could play a role down the line, although that's beyond the scope of the project. But what I want to highlight is in this project, not only we observed the formation of 2,4-dinitrophenol, we also observed nitrites, as I show in one of my slides, and that has never been reported in the hydrolysis work in the past because it proceeded under a different pathway. And because nitrite can be released, which indicates that less toxic product could be formed. And that, I think, is a highlight of this heterogeneous hydrolysis system, which seem to have the potential of favoring a different pathway, which will minimize the formation of 2,4-dinitrophenol. I hope that answers the question in a way.
Thank you very much. Great. So you spent a lot of time talking about DNAN hydrolysis, but this has been studied for a long time. What are the key highlights from your work that are novel?
Yeah, so you're absolutely right that DNAN hydrolysis have been extensively studied both experimentally and computationally. And on the experimental side, DNAN hydrolysis mostly forms 2,4-dinitrophenol, which is one of the main products. And as the previous question pointed out, is actually more toxic than its parent compound, which is problematic. The other possible pathway that was only being predicted in computational chemistry is the formation of a Meisen-Heimer complex, the other pathway which was predicted in computational model is the denitration pathway. Although this pathway was predicted to exist, but it has a very high activation energy, which explains why it has never been observed experimentally. But I think in this work, this is the first hydrolysis reaction reported in a heterogeneous system. In other words, the surface promoted hydrolysis. And we showed that the presence of surface was able to lower the activation energy, which can possibly contribute to the preference of a different reaction pathway that forms nitrite and less toxic products than the homogeneous hydrolysis pathway.
Great, thank you. This is a question of attendee. Do you expect the treatment system you described to be applicable to other contaminants, specifically compounds like PFAS?
That's a good question. So as many of the audience probably know, PFAS is very difficult to degrade. I would say there's some aspects of this project might be able to help concentrating PFAS. But in terms of degradation, I think that could be very challenging and require further investigation. Thank you.
Thank you. All right, you compared various PCM-like polymers in promoting DNAN hydrolysis, and you attributed the differences to their functional group identity and pore characteristics. What about surface area? Does it really matter in this case?
That's a great question. So let me kind of back up a little bit. Depends on the compound. If you have a compound that is very not absorptive, and I would say, yes, the first step of the compound migrating from the aqueous phase onto the solid phase is important. And the surface area would be very critical in that case. So to avoid the role of surface area in our system, we actually tuned the system to have all the contaminant loaded or preloaded onto the surface. So in that case, surface area doesn't matter because 99% of the compounds started out on the solid phase. And they can't, so basically at that point, this approach allows us to focus on the functional group identity and pore characteristics. Thank you.
Great, thank you. All right, another question. How can you be sure that the observed decrease in NTO and DNAN is due to transformation rather than adsorption?
So that's also a great question. My answer to that are two aspects. One is we actually looked at the total mass balance. So in other words, we looked at the solid phase and the aqueous phase, and all the results are the sum of the solid phase and aqueous phase mass. So we were able to track the solid phase down to understand if it's absorption or reaction. And on top of that, the formation of products would be another line of evidence to indicate whether or not this is reaction or passive absorption.
Great, thank you. Can your technology be combined with other technologies in a treatment train approach?
I would say yes. We currently have not looked into that, but hydrolysis is really a great approach in that if we think of our body, hydrolysis is typically also the first step we break down any complex molecules. And I would imagine it could potentially serve the same purpose in that it can transform a compound into an intermediate that could potentially be more hydrolyzable or prone to other microbial degradation or reduction processes. So the answer is yes, and I see a lot of synergies of this project that can bring to other existing SERDP projects on bioremediation, biostimulation, and potentially abiotic reduction. Thank you.
Thank you. How would PCM or PLPs be deployed in a remedial setup in the field? How would you transition the work that you're doing in the lab to the field?
So I will try to separate that question into two sides. One is the PLPs. So the PCM-like polymers is really a approach we're trying to use to understand what are the key properties. We currently don't have any plans to work on the scaling up process to deploy PLP. We're just trying to use that as a platform to identify key properties and translate that knowledge into PCM. So in terms of PCM, I think I gave a few examples of potentially modify the PCM surface. For instance, in this project, we demonstrated that the quaternary ammonium group is very important to accumulate OH- near surface. And then in one of my slides, I demonstrated that we can craft the QA groups onto activated carbon, and we did see an enhancement in facilitating the degradation of both TNT and DNAN. So down the line, we will look into other surface functionalization to really maximize its efficiency, and that can potentially help the transition into the field and deployment.
Wonderful. Thank you so much for a great presentation and a very engaging Q&A session, Wen-Cheng. At this point, I'd like to pull Craig back into the discussion and ask both of you some additional questions.
Sure.
Starting with a question for Craig. Craig, you talked a lot about putting nature to work, which I think resonates a lot with many of our audience members. Can you give some broad examples of how that might work with your proposed technology?
Sure, yeah, I did elaborate a little bit during the talk, but you know, what we found from our results is that, you know, more organic matter is better. Cycling through anaerobic and aerobic environments is better than just aerobic or strictly anaerobic, right? Because most of these compounds have both aerobic and anaerobic pathways for decomposition. And for NTO, for instance, the initial degradation is anaerobic, but then it kind of gets stuck unless you have aerobic decomposition pathways. So basically, either selecting ecosystems or modifying ecosystems to enhance, either organic matter abundance or this redox cycling. So for instance, like if you had a stream, it's similar to some approaches that are being used for nutrient management too, which is re-engineering the stream so it has more sinuosity, or you simply just put in pools and riffles so that there's more exchange between the surface water and the subsurface, which helps to create these anaerobic aerobic gradients. Pools themselves will act as organic matter traps, so you would also end up enhancing the amount of organic matter that's there. From a wetland or a treatment wetland perspective, the idea of having sort of adjacent reservoirs where you move water from a treatment wetland into a reservoir, let it dry out, and let it grow grass, for instance, and then pump that water back in. So now you have a ready supply of organic matter that is ready to fuel some of the decomposition reactions. And then you've also altered the redox conditions too. So you again achieve both things in terms of this redox cycling and amping up the amount of organic matter that's there, which would both contribute to enhanced mineralization. And then our organic matter amendment experiments show that low C to N ratio organic matter, so like phytoplankton or microalgae, you get more of a mineralization bang for the same amount of organic matter addition buck. So if you have some impoundment, that's got MC contaminated surface water in it, you could do nutrient amendments to that water to basically enhance the amount of microalgae or phytoplankton production. And when that settles out, it bumps up the organic matter supply of really good usable catalytic stuff for mineralization. So those are some ideas, from a hydrologic perspective, as well as sort of manipulating, the amount of productivity that's in the system.
Thank you so much. And Wen-Cheng, do you have something you'd like to add as it relates to your technology?
Yeah, absolutely. So putting nature to work is really my favorite topic. Basically, I think this technology lends itself to this capacity. The core of this technology is really building upon PCM, which one of the most commonly used PCM is activated carbon engineering. And that has been traditionally used as passive absorbent, and people have been adding activated carbon as soil and sediment amendments. And lately, people have been injecting colloidal activated carbon into groundwater for chlorinated solvents. So all of that are existing applications of activated carbon. And in this project, we actually reported something new, that the activated carbon is actually reactive in that we could potentially recycle those materials and elongate its lifetime in those environments. So one possibility is, using those tailored carbon for hydrolysis reaction, which occurs naturally at pH 7, and we don't have to do a lot of things on the sites. So I guess that would be a classic example of putting the nature to work.
Wonderful, thank you. And one last question for Craig before we wrap up. This is a question from Polytechnique Montreal. You considered sorption and mineralization as the two major paths of change for the compounds you study. What about transformation into other stable intermediates? And can you measure these intermediates with stable isotope analyses?
Yeah, that's a great question. To some extent, we purposely skipped over a lot of the intermediate organic derivatives. For that argument that I made that we wanted to be able to get a number that was strictly the rate of decomposition into inert end products that had no other regulatory thresholds. So that said, in all of the studies, what we do is we do measure the concentrations of intermediate, some of the major intermediate organic derivatives, and that varies depending on the compounds that we're doing. So that you'll see that that's contained in the final report for the legacy work, and it's also it will be contained for the IHE work. Yes, you can. The second part of your question was, can you measure the stable isotopic enrichment in their intermediate products? And the answer is yes. So you can measure it in the parent compound, and you can measure it in the derivatives, but you have to be a little bit careful. It's analytically tricky in the sense that some of the intermediates, you have to be mindful of what the enrichment is of the source material that you're using. So if you have a system that's set up for measuring, and there are a number of systems, all the compound-specific isotope analysis folks have some systems that'll measure RDX, a couple intermediates, TNT, a couple of their intermediates. But in very, very high enrichments, will kind of, I'm going to get in the weeds here, kind of swamp your column and cause you some analytical problems. But it can be done, and it might take a little bit of trial and error and paying attention to what those enrichment levels are. But yes, it can be done.
Wonderful. Well, thank you, Craig, and thank you, Wencheng, for a great webinar. We're going to go ahead and wrap up now, but before I remind you to join us on Thursday, November 17th for the next webinar in this series on DoD-funded research efforts to improve natural resource management by better understanding how disturbance and climate variability impact DoD lands. This webinar will feature two speakers, Dr. Scott Fehrenberg from the University of Montana and Dr. Sasha Reed from the USGS. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and other webinars through the beginning of next year. And before we conclude, I'd like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you'd like to refer to them in the future. We would appreciate it at this time if you can take a moment to complete the very short survey that will pop up on your screen. This concludes today's webcast. Thank you so much for joining us.